it was actually an interesting conversation that I had with Tarini recently when we were talking about a study around um, centurions, people that had lived, you know, uh, studying the microbiomes of people that had lived past the age of a hundred. And for me, that was fascinating because it was kind of the first time that I ever really contemplated the idea of the microbiome impacting the way that you age and even potentially the age that you may well live to. Is this an area that excites you, and where are we at with that? What is the thought around it at the moment?
Yeah, so I feel like a newcomer to this field, aging in the microbiome. But this last couple of years, we published a paper in this space because we had access to some data sets where we had a lot of data on these aging populations paired with microbiomes. And reading the literature, there was some contradictions that were out there, right? So there were a set of studies that were done in this cohort called ElderMet, which is I think a European cohort, and those people were older. They were largely living in assisted living facilities, so they were kind of not the healthiest of older people. They weren't community dwelling. They were no longer independently living on their own. They were sort of in these hospital like settings. And if you looked at their microbiomes, you found that over time, they're sort of maintaining a microbiome that sort of looks like a younger microbiome, but it's sort of losing diversity and changing slowly. And then it sort of collapses right before death, right? So as you get really, really sick and die, you see sort of a collapse of the classic structure of the microbiome. However, there were other studies looking at centenarians, these older people who live past 100, where they're changing. Their microbiomes are actually constantly adapting with age. And it looks like what's going on is that the core bugs, the core microbes that tend to dominate our guts when we're young, they're sort of losing dominance. They're lowering in prevalence, and all these other microbes that actually make us more unique, right? They tend not to be shared as much across individuals. These subdominant taxa are rising in prevalence with age. And this is a clear signal that's been seen across many populations of centenarians in Japan and Greece and other parts of the world. And so with our data set, we had sort of the ability to partition people based on their health state, these older individuals. And so we thought that perhaps what's going on is there's different signatures of aging here. One associated with less healthy and the other associated with maybe healthier people, assuming that centenarians are healthier than your average older person, which is probably a decent assumption. And yeah, that's what we saw. So we found that people who were aging in this kind of healthy population we had access to, we found that the abundance of these core taxa, specifically this genus Bacteroides that I mentioned earlier, it's declining steadily as you get older and older and older. And these subdominant taxa are increasing. We calculated a score to kind of quantify this. So we called it uniqueness. And what it is like it's just a score of how close are you to your nearest neighbor in the population? How ecologically distinct is your microbiome? And that score is rising with age, right? It goes hand in hand with the decline in core taxa. You get a rise in this uniqueness score. So then we took this other data set we had access to. It was called Mr. Oss. It was a cohort of older men, about 1,000 of them, who were being studied to look at osteoporosis in men, which is, I guess, an understudied phenomenon. But they had microbiome data and health data on these men. And when we looked at this uniqueness score with age, there was kind of a dampened association. We saw less of an association of this uniqueness increasing with age. So we thought, okay, what if we just partition these people by their health status? So we had a few ways of quantifying their health. One was walking speed. So if you walk faster as an older person, you're healthier. This is a well-validated metric. Another was drug use. So people who are on a lot of prescription drugs versus on not that many at all, that's another way to partition. And then a couple of other ways of doing it. And so however we looked at it, no matter how we partition people on their health status, healthy old people showed a really strong association between this uniqueness score and age. It's increasing as they get older. But people who were not as healthy, it was a flat line. So they didn't show any of this drift. Their microbiomes are staying kind of static as they got older.
And so help me understand this then. So what you're saying is going back to going throughout your life, we do see now a correlation between a certain category of, let's say, in inverted commas, beneficial bacteria, and if they stay relatively dominant, they can keep the bad inflammatory bacteria at bay. I know I'm talking like very broad strokes here, but so that if that carries through your life, you're, like we said, between the urban microbiomes and the rural microbiomes, a sturdy, good commensal bacteria microbiome keeps you in relatively good stead. But what you're saying is then, as you grow older, it's not these big sturdy microbes. There are these new, more unique microbes that come in that may play some kind of role in keeping you healthy as you get into latter years. And are you saying that commensal bacteria become less important?
It's curious, right? It's actually counterintuitive, this signature we're seeing. So yeah, like an 80-year-old person's microbiome who's healthy looks very different from a 20-year-old. You would think that a 20-year-old is like a healthy person, right? And their microbiome should be.
like that idea of transfusing a child's blood into you would think that, right? Like, I'll just get a transplant from a 20-year-old and then my microbiome is going to be where I want it to be.
And a lot of the field thinks that way, right? A lot of the microbiome aging field thinks that we should fecal transplant 20-year-olds microbiomes into 80-year-olds. And actually we're saying, no, hold your horses. Like, let's not do that. That actually could maybe do more harm than good. Because when we look at health status, we find that the least healthy people look more like 20-year-olds when they're older.